So thanks all for coming out today. I'm Frank Nothaft. I'm starting my third year in my PhD up at Berkeley across the bay. So I'll be talking today about Atom, a tool that we've been building to do high-speed, high-throughput genomic analysis on top of Apache Spark. I've tweeted these slides out, so I'm F Nothaft on Twitter. If you'd like to share them with your colleagues, please feel free to. So I don't know what the makeup of computational people versus biologists is out here tonight. Who here is a computational person? And then who here is a biologist? Great. So I think that my assessment that most people here probably don't know everything about genomics is probably a fair assessment. So I'm just going to give a brief background to what genomics is like. What are some of the analyses that we want to run? What are some of the algorithms that we're running here look like? Fundamentally, when we talk about analyzing genomes, our goal is to go ahead and come up with some understanding of the source code for life. So the human genome itself is about slightly over 3 billion letters long. It's split into, one might say 46 files, so where each file is a chromosome. So this is just a long piece of DNA. You get one copy of each chromosome from your mother, one copy of each chromosome from your father. And then inside this whole genome, this 3.2 billion character long file, 99% of places in the genome are identical between everybody in this room, more or less. It's kind of shocking, even though we all look very different, we all act very different, we all have very different personalities, we're actually 99.9% the same. So the changes that we're looking here, the changes that give rise to diseases like cancer or Alzheimer's, they're really needles in a haystack. But one of the things that is also kind of interesting to note is even if we're talking about one change in in every 1,000 places, when you're starting with a 3.2 billion character long file, that is still a lot of changes. So we've got a lot to crawl through here to find out what it is in these minor, these small edits, most of which are just a single character substitution that leads to, to us being, as a human species, so wide and diverse, and then what leads to the diseases that people get. And the way that we've been studying this, really this is a trend that's picked up in the past about 10 years, is through this abstraction called genome sequencing. So, everybody hears, genome sequencing is so hot, there's so much more genomic sequencing data that's being created now than 5 or even 10 years ago, and a lot of this is due to this revolution called next generation sequencing. So what we're doing, we take that 3.2 billion character long file, we replicate it a bunch of times, and then we cut it into small sequences. So, for those of us who are more on the machine learning, the statistical side of things, essentially sequencing is just a Poisson substring sampling process. We take your genome, we replicate it, we cut out these, right now most people use strings, these little string fragments that are between 100 and 250 bases long. And, we sample from that pool under a process that looks essentially like Poisson sampling. And, just using this process for $1,000, given a couple of days, we can go ahead and get a copy of your genome that is just, a bunch of these fragments, about 1.4 billion of them. However, as all of you are probably sitting here and saying, what am I going to do with these? You're telling me the genome is 3.2 billion bases long, and you're telling me that these substrings that I'm getting are 100, 250 bases long. What am I going to do with that information? The general thing that allows us to do this is this process that we kind of in the field refer to as genome resequencing. So, probably everybody remembers back in 2001, there was a lot of fanfare about the Human Genome Project. What the Human Genome Project did was it went ahead and spent $1 billion to go ahead and generate a really high quality draft of the human genome. So, we established earlier we're 99.9% the same. So the big thing that we're going to do with these little substrings is we're going to use them to calculate a diff. So, we're just going to calculate, figure out, where does this string map to the genome? Is it different from the genome at that place? Is it really different? Is it just some error that we saw? So once we've gone ahead and computed, once we've gone ahead and captured the sequence, we're left with two important problems. And one of them we have a fairly good handle on today. That's how do we actually go ahead and compute this difference. But the second one, and a bigger one, is what do we actually do once we've gotten these differences? What do we do to make meaning of these small edits that are sprinkled throughout the human genome? And so the way that we get started with calculating this diff is through this process where we align the substrings. So we take our bag of strings, we just place them to the genome, and then we use a statistical process to say, okay, well, so let's say instead of seeing an E here, I saw an A. It's a nonsensical word. There's no TIMAS, TIMAS. That's not a word that anyone knows. But is that a random error? Or did the sequencer just mess up when it read that base? Or is this actually a real change? Is someone, one of those youth, that are always going ahead and misusing words misusing words everywhere, giving rise to, crazy new vocabulary? Are they trying to change the word times into the word TIMAS? That's essentially what we're doing with this whole genome assembly process. We're doing this alignment, and then we're using a simple model. The model may even just be, again, for the statistically inclined, a model that assumes that there is some, binomial distribution of variation in the genome, and then, we just look and see, okay, well, given our error model, do we have enough evidence that says this is an error, or well, do we have enough evidence to say that this isn't an error, or do we think it's just something we can throw away, a fragment, something that was lost during the sequencing process? So what does this kind of leave us with right now? We have these tools, and we're going ahead and we're crunching the genome, but when you take 3.2 billion letters of text and you replicate it 30 or 60 times, you actually wind up with a lot of data. And to be more precise, if you're looking at the human genome, this about a 60x replicated human genome is about 200 gigabytes of data. So the way that we're processing this right now is in this long pipeline that runs all these stages cascaded one after another, where essentially each stage is it's some sort of an algorithm that's cleaning up what we see. And some of them actually even map to really common analysis steps that one might do. This mark duplicate stage, as its name implies, would map to a data deduplication stage that you might run in a common ETL pipeline. These recalibration, realignment stages, they're just doing some data cleanup. They're just going through finding systematic error that's in these fragments that we've sequenced and cleaning them up. Then, kind of as we run through the rest of our ETL-like pipeline, we're going ahead, running a statistical process, we're cleaning up the data, we're downsampling, and what we actually wind up with at the end, these variants that we have here that we can go ahead and analyze for whatever disease process we're looking for, that's about 100 times smaller than the data that we started with. So we might go from a 200 gigabyte input down to a 1 to 3 gigabyte output. But there's a few problems that we have with this pipeline right now. And actually this one that I'm showing you right here is a pipeline out of MIT and Harvard that's it's called the Genome Analysis Toolkit. The name is very straightforward and to the point. It's probably one of the most widely used tools out there, but it has a lot of people who detract on it because it's kind of slow. It's hard to tell what all these stages are doing. This whole pipeline to run end to end takes 3 days to a week to analyze. So you can imagine, I'm someone who comes in, I have cancer. I want to find out, is there a genetic cause for my cancer? Could I use this to pick a better treatment? You're going to have to get your genome sequenced and then spend 3 to 7 days waiting on the results of this pipeline. Additionally, if we look at it from the lens of what are the best practice tools out there in data analytics right now, a lot of these systems are relying on technologies that were commonplace when the sequencing trend picked up. The files that we're starting with, when we get this raw data in, some of the time it's even stored in just a pure text file. We might start with 600 gigabytes of data stored in a text file. We might have some of these binary files like BAM or CRAM that are used for storing genomic data. They actually have standards that are just hand implemented painstakingly by people. I was going ahead, we were playing around with some legacy data just yesterday. We were trying to make the reference Java implementation of the VCF, the variant encoding file format, talk to the C reference standard implementation, and we couldn't get them to talk because there's some slight error in how they process header files. And this is coming because there's some human sitting in a research lab somewhere who wants to analyze their genomes, but they've been told by their advisor that they need to work on the standards file, getting that to work, and hey, some human error slipped in somewhere. Additionally, these architectures that people are using are, they're very bare bones. They're very flat. One of the big abstractions that we rely on in genomics is that all data, most of the data that we're working on is aligned to a position in the reference genome. So, if we're trying to look, how does my genome differ from yours? We might say, I'm interested in the BRCA1 gene. I want to see if Frank's BRCA1 gene is different than someone else's BRCA1 gene. And we'll say, we know from studying the human genome exactly where the human genome is, exactly where the BRCA1 gene is, and we'll zoom in on that. The way that these legacy tools will let you process data is you normally get one abstraction, and it's just we give you a sorted iterator. You seek on the iterator until you hit the position that you're looking for. And I'm sure that programming against a sorted iterator is the level of abstraction that most of us would like to rely on in our day-to-day life. We just want an iterator. That's all that we get. None of these fancy map things, none of these fancy map reduce schemes that we can program against. So people actually really have to kind of contort their algorithms to fit this sort of really bare bones programming model. Additionally, one of the funny things that whenever I go and talk to biologists, one of the tropes that we have is, some of the time the most efficient way to process this data is I just go ahead and I'm processing 5 gigabytes of data that's in a text file format by processing it with sed, awk, grep on a command line. There are better abstractions to be using to do these ad hoc queries. We have nice tools like Spark SQL, we have Impala, things like Hive that we can use to do these queries. So these low-level abstractions aren't productive. We tend to use flat architectures that lock you to a specific computing architecture. Like a lot of these tools are designed for HPC farms with really expensive shared data storage. And then, one of the really subtle things is that when you're programming at this level, bugs creep in. You have to keep track of all of these, weird invariants about your data in your mind. You can't rely on a higher level programming environment to do that. And we've actually run across several places where these tools have introduced bugs just because, I was programming against a sorted iterator. I didn't think to check this weird invariant that I had in my data. That would have been trivial to check if I was writing, say, map reduce and then checking for something weird happening when I'm doing a reduce. So, about 2 years ago, we started thinking, hey, at the same point in time that this next generation sequencing got hot in 2006, a lot of things were heating up in the analytics space. I think the first production release of Hadoop was 2007. Spark came along 2010, 2012, and there's been a very similar growth in analytics as there has been in this genomic sequencing area. So we thought, hey, we're going to go ahead and we're just going to use the best of breed tools that are out there right now, try to define a new greenfield approach, try to build some of these common genomics analysis tools on top of that. And really the first thing that we did was we did what any classical database person would do and say, hey, can I come up with the schema for my data? And the big reason that we did this is that if you start to plan ahead, a schema provides a very nice narrow waist. So we actually use Avro. This is a slightly hacked version of our schema to make it a little bit more readable. But we use Avro for the schema, for defining the schema for most of our data. What's nice about this is that we can easily then process our data inside of Spark. We can process it inside of Hadoop MR. We can pull it into Spark SQL. We can change any of the way that we're actually doing our processing up here without having to refactor our schema. So once we came up with this schema, one of the big things that we realized is, well, we were talking about how there's a lot of processing in genomics relies on this abstraction where the genome is a coordinate space and we align to it. So we said, okay, we have Spark. Spark is pretty performant for most things. There's actually a lot of genomic processing that you can do with just map, filter, group by. But if you start looking at more advanced tasks, we can really accelerate some of them. So one of the big things that we did was we came up with specialized join strategies. So to deal with this genomic aligned data where we have data that is put at a certain point at a coordinate space, frequently I want to take one set of data and overlap that with someone else's set of data. So maybe I would take my genomic variants and John's genomic variants, overlap them, and see, is there a lot of commonality between these with where these where these variants hit in the coordinate space? Where we have areas where there's not a lot of commonality, we might then take a description of where genes are in the reference genome, overlap those, and see if there's any interesting results. And it turns out that if you're smart about how you use partitioning in Spark, if you're smart about the algorithms that you actually use for doing these comparisons, you can make this, we call it an overlap join. You can make it into a really efficient sort merge join. So we had a lot of fun hacking around on the algorithmic side to accelerate a lot of these common genomics patterns. Additionally, one of the big things that we did at an early date was we thought about what was actually the correct storage format for this genomic data. And there's a few a few interesting things. So genomics relies a lot on slicing and dicing of data. For certain algorithms, I only care about a subset of my fields. And that's very common. If you went back a couple slides, our schema just for describing these short genomic reads, these fragments that we're sampling, it has 33 different fields in it. But most analyses will touch a subset of maybe 8 of the fields. So we don't want to materialize the whole record whenever we do one of these analyses where we touch less than 1/5 of the fields in the record. Additionally, we commonly don't touch the whole data set. We might say, even though I have this whole genome, I'm just interested in a gene that's on chromosome 20 or a family, an interesting set of genes that are on chromosome 20. So we very commonly want to go ahead and only touch a subset of the data. So this actually matches really well with columnar storage. So we use the Apache Parquet format to go ahead and take our schemas and materialize them to disk in the Parquet format. And we've actually been doing a lot of work recently to try to improve some of the performance of both Parquet on disk and Spark in memory by one, using Parquet to actually store data on shuffles in Spark. So this can give us some compaction. We have some fields that have a lot of repetitive metadata. By storing it in a columnar form when we do the shuffle, we take up a lot less space when we're doing the shuffle. We pay a slight cost for going row-oriented to columnar, but that's the space savings are worth it. And additionally, we've been working on some optimizations to Parquet recently to try to improve the performance of some of the filtering code, which we rely heavily on. Sorry about that. And so one of the actually very interesting aspects of going ahead and saying, hey, we think this columnar file format, this columnar approach is better for genomics, is that we've actually gotten a bit of criticism from the genomics community that says, oh, well, you think our file formats aren't good, and here you come along, just like everybody else who has come before you, and you're going to introduce a 15th file format. Whose problems is this solving? So we actually had someone tweet this at us a while ago, which was kind of fun. But one of the really subtle points, and one of the great things about having chosen this schema-driven approach where we're using a schema as a narrow waist, is it doesn't matter what we're storing our data in as disk. It's actually very trivial to go ahead and say, okay, well, if I want to process legacy data, then I just define some way of going from this legacy file format to my schema. I just define some conversion that I can do. And this is actually really easy to do. So one of the things that's kind of great is that in the process of developing a flexible, fast, efficient processing stack for genomic data, we've actually developed a Swiss Army knife for converting between genomic formats. And everyone hates us for it. No, I jest. But kind of the simpler way of looking at it is that, if you put your database hat on, this is essentially a view. If you squint at it, this is essentially a view. So this was kind of one of the nice things that came out of this stack-based approach. So just to shift gears for a little bit, I want to talk about some of the work that we've been focusing on recently, which is how you actually use this, how you use this software stack to go ahead and analyze these genetic variants. So there's actually a very big challenge in this, where okay, so we know that fundamentally the differences in our genetic code drives the differences in our physical appearance, mental state, what diseases we get. However, commonly when we go ahead and we analyze your genome and we wind up with this, I guess we have a bag of 3 million variants at the end. A lot of these variants don't really have straightforward explanations. So really, what we wind up with is this challenge of how do we find what variant is important? How do we go ahead and say, okay, this deletion that occurs at base 300,000 in chromosome 10 is why you are 10% more likely to get Alzheimer's over the course of your life. And so the big question is, how can we actually go ahead and make sense of this variation? And ultimately, there's really two drivers. So, one is very simple. A variant can go ahead and break the product of a protein. So, if I have a variant in the body of one of my genes, it can actually change the outcome of translating that gene into a protein, right? So, it's essentially I change the recipe, I get a different outcome. But the other thing that's very subtle and that we've started looking at is that a variant that you have, even if it's not inside the body of a protein, can change how much of some protein in your genome is expressed. So I'll talk a little bit about this one later. But really the question is how do we turn these genomic variants that we have into a profile for what diseases you might have, what diseases a population might be susceptible to. And the big way that we do this is, it's kind of obvious in retrospect. It's just, through statistical modeling. And some of these are actually really simple approaches that we use. Like one approach that's very common is just to use a standard chi-squared test, where you essentially run an aggregation query over every single spot in the genome where you have a variant, and you say, okay, well, I'm going to look at people who have this variant, people who don't have this variant. How often do they have this trait or this disease that I'm interested in? And I'm just going to run a chi-squared test on that. Alternatively, if we have maybe a more complex trait, we want to look, let's say, at something that's a continuous thing, like human height, for example, we might go ahead and run a more complex model. So, we might run this aggregation query and run maybe a linear regression model on top of, as part of this aggregation. So this body of work is kind of generally known as genotype to phenotype testing. And one of the great things, again, with using Spark is that these sort of aggregation queries are a very natural map to the computational framework that we have. So there's a reason that Sparks became popular in machine learning, but these aggregation queries, or a lot of these aggregation queries are actually like fundamental ETL tasks. Even before you get to the machine learning to the machine learning side. So it's just a very good computational fit. But one of the problems that we run into really quickly, okay, so I did my statistical modeling, but something that's kind of a disappointing result is, well, very commonly I'm going to have these two changes. These are single base modifications. We call them SNPs, single nucleotide polymorphism. They're, maybe these two are 500 bases away from each other. And it turns out that they have the same probability value. Both of them are correlated at the same rate with me having a disease or having a change in how much of a copy of a protein that I make, something like that. And this phenomenon is called linkage disequilibrium. So it's essentially just a simple way of saying, what a lot of us know about locality, that, well, if, two things are close by to each other in the genome, they're probably going to get inherited together. So, even though, probably only one of these, at least for the rhetorical purposes of this talk, let's say only one of these variants, causes a certain disease. Maybe one of these variants makes you 5% more susceptible to diabetes. Because they're close to each other, they frequently get inherited together, and thus, we can't go ahead and figure out which one is actually the culprit. So one of the questions that I've been thinking about recently is, can we go ahead and break up these blocks? And, in some cases, this is easy. So, getting back to when we were saying, if the variant is in the body of a gene, then maybe we can predict how the gene is modified. And this is a common, common thing that people have done, where they'll say, okay, I have these 3 base pairs, I have this repeating thing of 3 base pairs that goes ahead and tells me what amino acid to make. So if I have a variant that is in one of these that changes what amino acid I'm going to make, say I modify an A to a C, that tells me, to make something completely different, maybe that's going to change what protein I actually get. Maybe it's going to change how the protein gets, spliced or folded together. So at least conceptually, when we have variants that are inside the body of a gene, if they don't modify the protein, if they don't modify, how the protein is spliced together, we can probably go ahead and discard it. So, if we knew, this protein didn't cause any functional change, this protein did, and they were in the body of a gene, then we could say, okay, well, SNP2, although you're highly correlated, you're not actually important. We could toss SNP2 out. But, this picture actually, kind of just a bit of biological, imagery nomenclature. This little arrow here implies that this is where a gene starts being transcribed into a protein. So these variants don't actually have any, they don't cause a functional change in what protein I'm getting. So the big question now is, well, it turns out that the part of your genome that actually codes for proteins is about 1% of your genome. It probably stands to reason that 99% that isn't actually, getting transcribed into a protein is probably important for something. It's probably not there just because, nature really likes replication, really likes leaving comments in its source code. And, just a little bit of credence for this kind of comes from a project that we've been looking at that's focused on acute myeloid leukemia. And what we have here is just kind of a decision tree based approach of looking at how, how mutations are acquired in this cancer. So, if you branch off to the left, that means that you did, so actually each one of these bubbles is a gene. If you branch off to the left at one of the bubbles, that means you did not have a mutated copy of that gene. If you branch off to the right, that means that you did have a mutation. The first thing that you'll notice in this chart is we have this huge long tail of people who have no mutations. And this accounts for a fairly substantial portion of people who have acute myeloid leukemia. So this tells us that these are the people who have no mutations in their genes, no mutations in the coding bodies of their genes, but yet they're still getting this cancer. And we know that cancer is a genetic disease. So what's going on here? So one of the big things that we've started doing is, there have been a group of people who are kind of like in the theoretical biology, systems biology community who have proposed essentially a grammar for what these modifications mean. So, you might say, okay, well, if I have a modification that occurs, let's say 500 bases before I start changing the sequence into protein, then if that goes ahead and changes how 2 proteins bind to the DNA to actually do the translation process, then maybe I'm not going to get any of that gene, or maybe it's going to modify the rate of expression of that gene in some harder to predict way. So we've been going ahead and building on top of Adam using a lot of these techniques, like these aggregation queries that we're running, these coordinate space joins. So we'll go ahead, run a coordinate space join to pick out these areas of interest. we'll then go ahead and run these aggregation queries to try to associate some level of significance with, with what we're seeing to try to understand this. And the big goal, what we're trying to get at the end of the day is we want to take this statistical model that gives us these, these probability scores, and go ahead and annotate it with more data. So, we want to annotate it with more data, add more data in, just keep on joining data in until we can fundamentally understand better, why is it that SNP2 is actually the causative, why is that the causative variant? Why is that the causative mutation and not SNP3, even though they're inherited together? So, that's what we're working on. There are lots of great ways to get involved in this sort of work if you're interested. For our group at Berkeley, we have a project called the Big Data Genomics group. We're an organization on GitHub. We have a variety of tools that we're working on kept there. All of our work is Apache 2 licensed. Some of our projects have actually very large numbers of open source contributors. Some of the time it's people who are in labs, sometimes people who are working at biomed corporations, and sometimes it's actually just people who are developers who in their spare time said, hey, this sounds like a cool idea. I'd like to learn more about this. And just said, hey, this is a small patch that I can make to help. So, we try to keep track of issues for all of our projects, and wherever possible, we try to tag them with pick me up if they're an issue that either doesn't require too much Spark knowledge or doesn't require too much genomics knowledge to get started. Additionally, if you're interested in this subject but want to get more of a tutorial, one of my colleagues, Uri Lasserson from Cloudera, put together a chapter in the Advanced Analytics with Spark book that just came out from O'Reilly a couple months back. It's chapter 10, and it just runs through an exact example of how you would use Spark to implement an example genomic analysis. So Uri's really interested in immunology. Before he went to Cloudera, he was an immunology PhD student. So his analysis focuses a bit on how you can use Spark to understand the immune system a bit better. Additionally, we had a paper come out on Atom just back in June, or just back in May. So we have a lot of the more technical details on Atom in that paper. And additionally, I'm involved in a standards organization called the Global Alliance for Genomics and Health. I didn't talk about this much in today's talk, but biology faces a big problem where data is frequently stored in silos, and data sharing is very difficult. So this standards body is created with the sole goal of coming up with computational solutions that allow it, that make it easier to share data. One of the projects that they have that they've been really looking for software engineering help on is this project that is their server implementation. So it's just a way that people who, maybe I have a small lab, I don't know a ton about computers, I just want to go ahead and set up a simple server that I can use to share the data that my lab is collecting with people at other labs. It's actually, I believe it's just a Python-based web server, so they're always looking for people to contribute code. The code base is fairly clean, fairly simple, fairly straightforward to get started on. So these are a few good ways that you can kind of jump in and if you're interested in kind of the computational open source environment in biology. So thank you all for listening today. I hope you found it interesting. I would just like to thank although I am standing in front of you, this work is the product of many people's labors. We've actually had over 40 people who've contributed to the Atom project at more than 12 institutions. So I just want to thank my collaborators. Thank you.
We certainly have time for questions, so just ask Frank to repeat the question for the recording.
Definitely. Sure. So the question was, can I talk about how Atom relates to the Google Genomics platform? I know Google has been working on their Genomics API for about the same amount of time that we have been, and so they have several different APIs, some of which I'm more familiar with than others. The caveat is that my information is a little out of date. But so one of the things that Google has tried to make fairly easy to do is they've tried to make a set of kind of pre-canned common analyses available and easy to run. So there's a large bevy of common analyses like variant annotation, trying to annotate these causal effects onto the variants, or trying to go through and turn reads into variants that they've made easy for people to submit their codes, or easy to submit their data to through kind of a combination of a web-based interface with a, I guess with a tie-in to data storage on the Google Cloud Engine. For Atom itself, our tools are less platform dependent. Obviously we rely on Apache Spark, but other than that, we're not tied to the Amazon cloud infrastructure. We've actually run it on standalone clusters at our side and at other institutions, Amazon cloud, the Azure cloud. So we're very portable from that sense. Our APIs are a little lower level. If I wanted to kind of capitulate what Atom is as a tool, Atom is actually the first level of an API for going ahead and defining a lot of these genomic queries. So it's very general, and then we've built several tools on top of it to implement these algorithms. It's like for me, I spend a lot of time working on Avocado, which is our variant calling pipeline. We have several other projects that are focused on doing gene expression analyses and whatnot. So I think they're similar technology, or similar stacks. I think the Google stack under the hood relies on many technologies that are similar to what we're doing. I've heard a lot of really wonderful things about people who are using it, especially to analyze variants. I just haven't played around with it a ton myself. Oh, sure. So the question was, on this slide when I was talking about cancer data, I implied that the data was very homogeneous, when most of the time cancer data is said to be very heterogeneous. I think I had misspoke when I was talking about this. So my point is that we have this long tail here that we can't really explain through conventional variation, like variant analysis, like variant annotation methods. But actually, there are several clusters in this tree that drop out into very clear heterogeneous clusters. So if you go in, there's, like off the top of my head, there's about 8 different subtypes that go ahead and correlate with well understood mutational groups. Like some of the common ones are like there's a FLT3 internal tandem duplication that has two different subtypes that associate very clearly with it. My point was just that there's this long tail here that our conventional methods don't really explain because they're capturing non-exonic variants. Yeah. So the question was, is this patient data or from the cancer itself? So this data is from the TCGA AML cohort. So it's about 200 patients that were sequenced at, I think, mostly WashU in St. Louis. The data is published, so we actually, this data, I can't remember if we got it actually from the mutation calls or from their spreadsheet that annotated the mutation calls with the genes. But it is from real patient data, and it's from the mutation calling pipeline. So these are the mutation calls. Got it. Yeah. So to be a bit more specific, so the way that this data was obtained, so this is a diff actually from the germline against the cancer cells. So what they did was they sequenced I think skin cells for the germline, and then they sequenced the blood cells for the quote unquote tumor. It's AML, so it's not really solid tumor. And then the variants that they've identified here are the variants between the non-mutated cells and the mutated cells.
So as a newbie to this space, but with a significant amount of experience in Scala and other software platforms, how can we contribute in a meaningful way?
Sure. And this was actually something I was talking about with Alexi earlier. So the question was, if I'm a newbie coming in, I don't have much biology knowledge, but I have a lot of computational knowledge. I have a lot of experience with Spark, a lot of experience with Scala, something like that. How can I go ahead and start contributing? And so there's a few answers. One of them, actually, we've had a lot of people who come in with computational knowledge, but not necessarily the biology knowledge, who say, oh, well, I'm going to start out by, maybe I'm going to go through and I'm going to find one of the algorithms that you're running, I'm going to profile it, I'm going to put in some performance improvements, and then go ahead and, that's how I'll learn my way through, what this data looks like, what are some of the algorithms that people are running. And while that sounds like a bit of a joke, we've actually had several people come in. I think we've got, in one algorithm, we got about a 30% speed improvement out of a person who just came in from the open source world and was like, oh, I don't know much about genomics, but I know a lot about profiling code. And they got us about a 30% speed improvement in one big algorithm we run, and 10% in another one. So that's one place, and especially for us, we realize that there are a lot of things where it's easier to get into some parts of the code base if some parts of the code base are easier to get into without too much biology knowledge. So when we have our issues, when we post them on our GitHub, we try to tag those with, hey, you can probably get into this one without knowing too much biology. On the other side, some of the while some of the algorithms are really some of the algorithms are really difficult to play with. There's a few things that people have just been like, oh, well, I'm just going to look at this data with algorithms that I'm already familiar with. There's a few people who've come in from the open source world, people coming from the software side without much biology expertise, who are like, okay, I know in biology I want to understand how these variants correlate to populations. And I know how if this was normal, quote unquote, normal data that came out of an analytics pipeline, maybe I would go ahead and try to figure out how this data is segregated by running a k-means model. Or maybe I would figure out how it's segregated by running some other type of statistical model. So there have been two people who've come in from the open source world who've been like, oh, I just kind of want to get my hands on the data. What happens if I apply some of these statistical methods that I already know to the data, just played around with it a little bit? And that's been kind of a way that people are like, okay, I'm going to bite off a problem that sounds like kind of reasonable and just do some exploratory data mining and see what happens. And it's actually led to some pretty cool work. So we have a few blog posts, if you go to our website, bdgenomics.org, that are from people who've done that and who explain what they've done. So.
Maybe a little bit different approach. I'm considering taking a couple online courses. One at Johns Hopkins taught by Ben Langmuir, and another UC San Diego, which they keep running into. So I'm thinking, because they're going to teach us pretty basically, and they're going to run with Python and learn just these algorithms. But what I'm curious about is what type of link or then connection, because I've been studying Spark as well, I'm doing the edX series right now. If I will, on my own, let me try this with Atom now, or Spark, if I'll recognize that translation, or if I would also be able to get a little bit of assistance and feedback by joining in some of the discussion online and say, I recognize I'm studying this algorithm and I'm thinking about trying it out with Atom, this way, would I do this, or ask for a little advice.
Sure. So the question was, okay, so I'm interested in biology. I'm going to take an approach, I'm going to take some courses on edX or Coursera or something to try to get some knowledge of the space that way. How easy will it be to take the knowledge that I've gotten from these courses and translate it to the genomics area? And this is a really great question. I'm glad that you brought this up, and I'd actually really like to highlight one of the MOOCs that you mentioned. There's a professor at Johns Hopkins, Ben Langmuir. He's one of the most highly cited people who are about his age in genomics. Does awesome, incredible work. Actually, he actually does some of his own work on top of Hadoop, actually. And he's teaching a MOOC, I guess, right now or starting very soon. So yeah, I know a lot of the people in the genomics area are really excited for that. And getting back to your question, getting back to the point more, that's actually how a lot of people who are in our group well, our group, broader, a lot of the kind of people who are at Berkeley who come over from the computer science side have gotten into this. So, one of my colleagues, Matt Massey, he was the person who actually started Atom. He's the first person who committed any code for it. That's how he picked up a lot of knowledge about genomics. The two of the people who are the drivers on this AML project, actually, one of them is a computer networks researcher, and the other is a computer file systems researcher. He actually cut his teeth doing like mock kernel design, like back when mock was like the cool thing in kernels. They learned most of the stuff that's led to the work that we've been doing in the cancer space from watching MOOCs actually. So I think it takes a little bit of creativity to translate MOOC into algorithms, but especially like some of them, like the Ben Langmead course, I think will have a very straightforward transformation or a very straightforward translation. But yeah, no, it's definitely a great way to get started and to understand some of the background more.
It seems that this platform is mainly developed for eukaryotic genomes. Is it also handle prokaryotes?
Sure. So the question was, the platform that we're talking about is mostly designed for handling eukaryotic genomes. So genomes of humans, so on and so forth, versus prokaryotic genomes, which are like viral genomes and so on. So I haven't done any analysis of viral genomes with it. Conceptually, there's no reason why not. And like one of the big things that I kind of get the sense in the eukaryotic genome space is that there's a lot of people who want to, instead of going with alignment-based approaches, they want to go with assembly-based approaches. So I've actually been doing a bit of thinking along with some other people in our group about how we can efficiently support assembly. One of the processes, we talked about the Human Genome Project many slides back. What the Human Genome Project did was, since they didn't have a human reference genome, they went ahead and they assembled one from scratch. And a lot of these algorithms that you run to go ahead and assemble a genome they're graph-based. So we've actually been doing a lot of work using GraphX, which is a Spark library that provides efficient graph processing support on top of Spark to do these sort of genome assembly tasks. So the short answer is, I don't know if anyone's pushed that sort of data through it yet, but I believe that long term it shouldn't be something that we can't support.